Hello, I'm Dr. DeBellis. Today we're going to be talking about the anatomy of neuron. As you're aware, our brain is a really complex organ that allows us to reason, remember, understand things. This wouldn't be possible without the neuron. The neuron is a specialized cell that's capable of sending a signal. Now, one signal isn't going to make all the difference in the world necessarily. However, when we have assemblies of these signals, this is what gives us the ability to understand other people's perspective or abstract concepts. So the neuron is essentially the building block of the brain. One of the foremost experts on the neuron was Santiago Ramon Cajal. There was a time when we did not understand the significance of neurons. There were a lot of technological advances that gave rise to our understanding, such as the textile industry, which created many different types of dyes, which allowed us to actually study neurons under a microscope. Cajal came up with the neural doctrine, which is the hypothesis the brain is composed of separate cells that are distinct structurally, metabolically, and functionally. And information must jump across gaps between them. This was an innovative idea that there were gaps between the neurons. Prior to that, it was assumed that neurons were just connected like wires. It was ideas like this that allowed Cajal to go on to win a Nobel Prize. He's a very interesting individual with many gifts. The illustration on the right is actually one of Cajal's. He was also a medical doctor as well as a medical illustrator. Now we just talked about a neuron. And again, a neuron is an electrically excitable cell that processes and transmits information through electrical and chemical signals. This is different from a glial cell. A glial cell is non-neural. It's a non-neural brain cell that provides structural, nutritional, and other types of support to the brain. So it doesn't have the ability to carry an electrical charge typically. It's not electrically excitable, we would say. Now, we're going to say that we have 4 different types of glial cells. You actually do have some that I didn't talk about in here, such as radial cells and endothelial cells, but we're going to stick with these 4, and I gave you an acronym down at the bottom. SOMA, this is an acronym for astrocytes, microglial cells, oligodendrocytes, and Schwann cells. And those are going to be the 4 types of glial cells that we discuss in this chapter. Astrocytes are so named for their shape. They look like stars. They're star-shaped glia that add structural support. They can communicate, and they can also swell. Microglial cells, named microglial because they're so small, they remove debris. They also have a role in pain as well. Oligodendrocytes and Schwann cells form insulation that wraps around the axon. And we're going to discuss what the axon is in a moment here. But oligodendrocytes are the glia that form the myelin sheath in the central nervous system. If individuals start losing this myelin, which happens in many different conditions such as multiple sclerosis, it can actually affect their cognitive processing as a result. Now, Schwann cells also form myelin, but they don't form it in the central nervous system. They actually form the myelin around the axon of the neuron in the peripheral nervous system. So if we're talking about myelin or insulation that's in the central nervous system, we're talking about oligodendrocytes. If we're talking about it in the peripheral nervous system, which is outside of the brain and spinal cord, then we're talking about Schwann cells. Now, the presynaptic region of a synapse that releases a neurotransmitter is going to be called presynaptic. So we can see here, this would be the presynaptic membrane right here. And neurotransmitters actually are released from the presynaptic region, and they're released into what we're going to call the synapse. And here we can see neurotransmitter in the synapse. And then the neurotransmitter actually diffuses across to what we're going to call the postsynaptic region, or the postsynaptic membrane. It's a region of the synapse that receives and responds to a neurotransmitter. Now, the presynaptic region is typically on what we're going to call the axon terminals. We're going to talk about what that means in a moment here. The postsynaptic region is typically going to be on these little dendrites here. We call these dendrites. They look like little branches on a tree. Now we can divide a neuron into 4 functional zones. First, we have the input zone, which we were just talking about a moment ago. So the input zone is going to be everything to the left of this line. It's going to be the dendrites that receive incoming information. They receive input from other neurons, and that typically is going to be through neurotransmitters that again are released into the synapse. We also have the integration zone here. The integration zone is where the spark of life that we're going to call the action potential, where it actually initiates. So the integration zone's the cell body information that combines info to determine whether an action potential is going to occur. And it typically happens in this little cone-shaped area that we're going to refer to as the axon hillock. That's really important to know, that the action potential is initiated in the axon hillock. And we're going to talk about this in great detail when we get to chapter 3 and we discuss neurophysiology. So the third functional zone is what we call the conduction zone. And the conduction zone is going to be between the 2 red lines. In the conduction zone, we have the structure we call the axon. And the axon carries information away from the cell body. Again, this right here is the cell body. And this right here is the axon. So the axon is this insulated wire that originates at the cell body, and it conducts a message to the output zone. And the output zone is comprised of the axon terminals, and they transmit the neuron's activity into the synapse, because we actually have those little neurotransmitters being produced, and then those neurotransmitters are then released into the synapse. And they're released from this output zone. And again, the output zone is going to be in the axon terminals. Now, I said that we have 4 different types of glial cells. We have the Schwann cells, the oligodendrocytes, the microglia, and the astrocytes. Whenever it comes to neurons, we actually have 3 different types. We have multipolar neurons, bipolar neurons, and unipolar neurons. Now, the multipolar neurons have many dendrites in a single axon. We just looked at a multipolar neuron. And that's what we're going to use as our standard garden variety neuron for this chapter. Bipolar neurons have a single dendrite at each end. And unipolar neurons have a single branch that leaves the cell body and extends in two directions. One end's the receptive pole, and the other end is the output zone. So we have all different types of neurons. We're mostly, though, going to talk about multipolar neurons. I also wanted to point out we have motor neurons, sensory neurons, and interneurons. Motor neurons stimulate muscles. That's why they're called motor neurons. Sensory neurons respond to environmental sensory stimuli, whether it's sounds, sights. And finally, interneurons receive input from one and send input to other neurons. And most of the neurons in the brain are interneurons. You can think of the brain as this collection of interneurons that are all competing with each other to process information. Here I have a picture with oligodendrocytes and Schwann cells. Again, oligodendrocytes are the glia that form the myelin sheath of the central nervous system. Here's an oligodendrocyte right here. Right, there's an oligodendrocyte, and then we have some Schwann cells as well. And here's a Schwann cell wrapping that axon. Again, a Schwann cell's glia that form the myelin in the peripheral nervous system. So now my question to you is, what's wrong with this picture? So go ahead and take a moment to look this over and decide what's wrong with this picture. And I'm not looking for anything about the style of the illustration or anything like that. There's something fundamentally wrong. You can figure that out based on what I've taught you already about glial cells. So the correct answer here is that we shouldn't see a Schwann cell and an oligodendrocyte next to each other like this, because the oligodendrocytes are the myelin sheath in the central nervous system, but the Schwann cells are in the peripheral nervous system. So having oligodendrocytes both before and after a Schwann cell actually is something that we probably would not see. So that's what's wrong with this picture. Now, myelination is when glial cells wrap axons with a fatty sheath, myelin, to enhance speed and intensity of the action potential. So they magnify its strength as well as its speed. The brain is really, really remarkable in the sense that it's capable of working on so little energy. Now we have calculators, and calculators require more energy, but here's the catch. One advantage a calculator has is that a calculator can typically calculate things at a quicker rate than the human brain. The unique thing about the brain is how little energy it actually takes to do the complicated things that a calculator is not capable of doing. So if we look at this illustration here, we can see the myelination. So this is myelination. If this is in the central nervous system, this is going to be a collection of oligodendrocytes. They look like beads on a necklace. So right here, myelin. Interestingly, we also have these little gaps, these little tiny gaps where the axon's not covered by any myelin. And that's no accident. It's not sloppy work. It actually serves a purpose. These gaps are referred to as the nodes of Ranvier, and they serve a purpose in ensuring that the action potential is conducted at the optimal rate. So nodes of Ranvier are the gaps between sections of myelin where the axon is exposed. Now the cell body and the dendrites receive information across synapses. So this is what we would call the cell body here. And these are the dendrites. As you can see, the dendrites have this branched arborization. When I drew these dendrites, I was actually thinking about some of the young saplings in my backyard growing. I was thinking of these small trees. I was thinking of their branches. You have these little tiny dots here that look like berries to me. These are actually called dendritic spines. They increase the surface area of the dendrites, therefore increasing the functional capability of the dendrites. So these dendritic spines are studded on the dendrites. Again, we can draw them here. These dendritic spines are studded on the dendrites and increase their surface area, their number, and their structure to be rapidly altered by experience. These dendritic spines can actually change their location in as little as a few seconds, giving rise to a phenomenon that we call neuroplasticity. Neuroplasticity refers to the fact that the brain changes itself. It changes in response to the environment. You're probably aware of this. You've seen people who've maybe suffered a stroke, and they have a loss of ability to move half of their body, typically their arms or their face, sometimes their leg. But as a result, they have limited motor function. However, with rehabilitation, oftentimes they can regain a significant amount. This is because of neuroplasticity. Now, the dendrites are studded with these small spines to increase their surface area, and these spines are constantly changing their arrangement within seconds, allowing for this neuroplasticity. And neuroplasticity is the ability of the nervous system to change in response to experience or the environment. As I pointed out earlier, this cone-shaped area right here is what we're going to refer to as the axon hillock. It's a cone-shaped area of the cell body that gives rise to the axon as well as the action potential. And when we get to the next chapter, chapter 3, we're going to discuss how this is actually, because this is the first area on the neuron that actually has voltage-gated sodium channels in its membrane. It's helpful for you to keep in mind, and I don't know that I've pointed this out yet, information is flowing from the dendrites to the axon terminal. So information is flowing from left to right. And once again, we are going to call this the input zone. This is the integration zone. We're going to call this the conduction zone. And finally, we're going to call this the output zone. The presynaptic membrane is on the axon terminal of the presynaptic neuron. So in this particular illustration, information is going to be traveling down. So this is the presynaptic membrane, and inside the presynaptic membrane, we're actually going to have these things we call vesicles, or these little pouches, and these pouches are full of neurotransmitter, and the neurotransmitter then is going to be released into the synapse. So there's the release of it. It goes into what we call the synapse or the synaptic cleft, which is going to be a gap that separates the pre- and post-synaptic membrane. Now the postsynaptic membrane is going to be right here. It's typically the surface of the dendrite of the postsynaptic neuron. And there we're going to have all these little receptors. These receptors can actually be activated by the neurotransmitter that's released into the synaptic cleft from the presynaptic membrane. Here we have another illustration of what this is going to look like. So top, you can see that vesicle there. And the vesicle, it's comprised of the same material as the membrane of the neuron. And we're going to learn it plays several really important roles. The first important role that it plays is it protects this from these little enzymes. We actually have all these little enzymes that are inside the neuron. And if you did not have this vesicle, this neat packaging, those enzymes would actually eat up the neurotransmitter. Apparently the neurotransmitter tastes really good because these enzymes really want to get to that neurotransmitter and consume it and destroy it. So the vesicle protects it, and the vesicle also allows the neurotransmitter to be safely released. The way that this actually happens, and we're going to discuss this in the next chapter, calcium actually enters the neuron and it causes the vesicle to fuse with the membrane. And through a process we're going to call exocytosis, it releases this neurotransmitter. And here's that neurotransmitter. And we have many different types of neurotransmitters. Typically we talk about the biogenic amines such as dopamine, norepinephrine, of course serotonin, as well as acetylcholine. But these are actually released and then they float along passively through diffusion. And if that neurotransmitter is the specific type that fits into one of these receptors, the same way that a key fits into a lock, it will actually open up one of these receptors. And then you end up with this gap right here. And a positively or negatively charged ion, called a cation if it's positively charged, or an anion if it's negatively charged, will actually be allowed to pass in. So one of these. Now the neurotransmitter then will oftentimes leave, or sometimes it can even be destroyed. But the postsynaptic membrane contains receptors, which are specialized protein molecules that capture and react to molecules of neurotransmitter. Here's another picture of that synaptic vesicle. Again, these are small spheres in presynaptic axon terminals that contain molecules of neurotransmitter. Then they fuse with the membrane, thanks to calcium. And then the neurotransmitter is released from the presynaptic membrane into the synaptic space. So neurotransmitters are released into the synapse in response to electrical activity called the action potential that causes the vesicle to fuse with the membrane and release neurotransmitter into the synaptic cleft. Now, molecules of neurotransmitter generally do not enter the postsynaptic space. They simply open up the receptor and allow charged molecules, again cations or anions, to enter. And this causes a slight graded potential, a slight change in electrical potential of that dendrite. So if we compare axons and dendrites, we can say that for one of these multipolar neurons, typically we have few axons. They're uniform in their thickness, kind of like a wire. They have a hillock, which we talked about as the spot where the action potential begins. Also, they're myelinated. In the central nervous system, they're myelinated with oligodendrocytes. In the peripheral nervous system, they're covered in Schwann cells. And axons are typically longer. They can be quite long, actually. If you think about it, in a giraffe, there's actually an axon that's going to travel the length of the giraffe's neck. It's going to travel down its leg. It's going to travel to its hoof. So this is going to be a lot longer than a dendrite. Now, in a typical multipolar neuron, we will have more dendrites than axons. And the dendrites are tapered. They don't have a hillock, and they're not myelinated, and they tend to be shorter than axons. So what I'd like you to do as we end this lecture is I would like you to draw a neuron and label the dendrites, the dendritic spines, the cell body, the nucleus, the axon hillock, the axon, the nodes of Ranvier, the myelin, the axon terminals, and the terminal buttons. Well, as an illustration that I did, I enjoy drawing it, maybe a little bit too much. You don't have to spend too much time, but I'd like you to be able to draw those so that you can solidify what you've just learned about the neuron. And when you're finished with that, you can look to see how you label them. There's the labeling job that I did on my neuron. As you can see, here are the dendrites like branches. And then on the tips of the dendrites, we have these dendritic spines. This is the cell body here, also called the soma, and it houses the nucleus of the neuron, which holds the genetic information. And here we have the axon hillock, which is where the action potential originates, where that spark of activity occurs, that spark of life that gives rise to the action potential and travels down the axon. And here we have the axon, and it's covered with this phospholipid here that we're going to call myelin, which increases the speed and intensity of the action potential. You have these little gaps, don't forget those, the nodes of Ranvier. We're going to talk about what the purpose of those is in the next chapter. And finally, at the end of the axon, we have the axon terminals. So we can call these the axon terminals, and at the end are the axon buttons. And these axon buttons are actually, well, terminal buttons we'll call them. These terminal buttons are actually going to release the neurotransmitter into the synapse so that it can go from the presynaptic space into the synapse and diffuse across to the postsynaptic space. So hopefully we can talk about this when we get to chapter 3.